Start sharing.
Yes, thanks. Can everybody see this and hear me?
We can hear you. We can see your slides. So take it away.
Great. Thanks so much. I'm very honored and excited to be a part of this. As you all just highlighted, there's so much data and discovery going on here, and our effort is really a testament to all this collective work. My project did not include any new data. It's getting maximal resources together in one place. And that's one of two big pieces I'll be talking about today, which is the public forward-facing NeMO analytics system, where we have worked together. Yash, the first author, pulled together a ton of data here. He couldn't speak today, so I'll be explaining things. Across mouse, monkey, and human, both in vivo brain development as well as in vitro models, and a good bit of adult data as well. And the idea behind the NeMO analytics is really to get gene-level data in the hands of as many investigators as possible. And, oops, sorry. My slides are working a little slow here. I apologize. Okay. So the first part is the public database, where anybody on the planet can type in a gene name and look at its expression across all of these systems. The other big piece here is that you can look at gene signatures across all these data sets that are all in one place. The second piece I'll be talking about today is actually how we generate some of those interesting gene signatures. So we take subsets of the larger collection and do joint decomposition that takes apart, instead of one data set at a time, multiple data sets. So the components you come out with are not a PC1 of one data set, but a component that is shared across many. And then we can take those signatures back into NeMO and look at them across species and times to see where and when they occur. So what does all this mean? This is an example of that decomposition. We took a mouse, a monkey, and a human data set from mid-gestation, all single cell data on excitatory neurogenesis. So the rows are those three data sets. The four columns are four shared conserved signatures that we have across the mammalian family tree for this neurogenic trajectory, moving from radioglial cells to IPCs to a very transient nascent neuronal signature, and then into signatures that are common to all the different kinds of maturing neurons in the different systems. So with this as a starting point, we can then go back into NeMO with these signatures of different stages of neurogenesis and learn more about them by looking at them in different data sets. So the first data set is some laser capture followed by RNA sequencing, and these are the same four signatures we were just looking at. So looking at the dissection of this RNA-seq data set, you can see the radioglial signature is highest in the ventricular zone, with IPCs emerging in the inner subventricular zone just as they delaminate from the neuroepithelium. The newborn neuron signature is actually still in the subventricular zone. These neurons haven't even moved yet. And then by the time they get to the cortical plate, they're expressing this mature signature, and that nascent signature here is not even in the cortical plate yet. So here we can add a spatial perspective to the signatures we got from single cell data that had no spatial information in it. And further, here's some even higher resolution data, and again, a testament to the kind of data that can be produced these days. This is stereo-seq from the developing mouse brain at E6.5. And again, these same four signatures that we got from the single cell data now projected into the spatial transcriptomic, so we can see exactly where these signatures are, with the radioglia lining the ventricles, IPCs moving just off that as they delaminate, and then the nascent and maturing neuron signatures as they radiate out toward their final resting place in the cortical plate here. So those are two spatial examples. One more is a data set that had used flash tag techniques to birth date neurons, or progenitors actually, before they were sequenced. So you know the number of hours since the cell's terminal division when you sequence it. So this allowed us to look at our signatures and in the mouse cortex actually track how many hours it takes cells to move through these sequential states of neurogenesis. And this sort of puts everything together. It's an older data set, but incredibly beautiful from the Allen with thousands and thousands of laser captures with the progenitors and the ventricular zones starting low in the nascent and maturing neuronal signatures. As those neurons are born here in red and blue in the subventricular zones, the nascent signature goes up. And very quickly, as those cells begin to move and mature, moving through the intermediate zone to the cortical plate, they turn off that nascent signature and turn on the maturing neuron signature. And we'll really get into this deeper now from another perspective. So looking at these mature neurons, we're going to start from adult data and go backwards to see where transcriptional signatures of adult mature neurons appear. So we grabbed a bunch of single nucleus data from the Allen, decomposed it. Again, only glutamatergic neurons here, so it's going to be all sorts of patterns of adult human neocortical neurons. And what this sort of overview image shows is that there's 9 rows here, each of which represents a laminar specific neuronal signature in excitatory neurons of the adult human cortex. Every column is another spatial or single nucleus data set from mouse, monkey, or human where we can observe them. So these are incredibly robust and identifiable across technologies and species. I'll just zoom in on 2 of them here. This is a layer 4 signature, and on top a layer 3/4 signature. The first 2 columns are spatial data from the human, and the signatures we got from those single cell data sets you can see are sort of incredibly precise in where these neurons actually came from. But of course, we wouldn't have known that from the single nucleus data. We can localize them with the spatial data. You can also see them here in the macaque, a beautiful sort of ribbon of cells in a particular layer as it snakes through the cortex. In mouse, we can see the same thing here with the layer 3/4 signature right there. It actually shares this signature with a bunch of neurons of the thalamus. And what's very interesting here is that we see the layer 4 signature is entirely absent from the mouse cortex. So we actually confirmed that in many other mouse data sets that it's primate specific. From a previous publication in the Nad Sestan's lab from 2022, we actually confirmed that this primate specific signature had already been discovered. But this is really just to give you an idea of how much real estate, scientific data real estate you can look across when you combine the data collection with transcriptomic signatures of interest. And now this is my final notion here, is to put these things together. So this is a data set from gestational week 24, watching progenitors as they go through neurogenesis, turning on the nascent signature into the mature signature. And then what's turning on there when those cells end up in this mature state is the color codes for one of these neuronal signatures in the adult human. So looking across all the ages in this Ramos 2021 data set, we can see that at all stages we're seeing neurogenesis happen, but just when we see this color is when neurons are of this particular layer beginning to show their laminar specific qualities. And we can do that across all the ages and layers. And then these green arrows are showing at what times the specific neuronal layers are coming up. And just as the birth order follows the inside out layers, so does their maturity, with a notable exception. A particular type of neuron are born early, and we show this in the paper. And finally, in my last slide here, just a moment to mention that we can do this also in organoids, looking for these patterns. And it turns out that despite the fact that almost all of the layer defining TFs show up in neurons of organoids, only very few of the full adult transcriptomic signatures show up as they should. And in this time course from the Canton paper, we see that these 2 deep layer neuron signatures show up. But if you put organoids in the head of a newborn mouse, you can get almost the full complement of mature neuronal signatures, notably lacking the primate specific one that we identified. And again, hopefully these tools will be really used across the planet because you guys and many others have produced all this data. We're just trying to put it in the hands of as many people as possible to do some really creative